Hello everyone, in this video I am going to do recap of lecture 1 of Arnab Gupto sir in gene regulation, that is LS4203, and the lecture was on 4th of March 2024 and I was absent in that, but I Shodipa discussed with me about the lectures, so from her notes and from our discussion I recap this. So first of all, what he said that there are three parts which he's going to teach us. First of all, cellular organellar contacts, and he will be telling us how to check that two organelles are contacting each other, and then condensation, condensates, CONDENSATS. So these are some dense portions of the cytoplasms. The cytoplasm is not homogeneous, and there are certain portions which are denser than the other portions. These are called condensates, and I do not know what he is going to teach in this. And then he's going to teach us signaling, something about signaling. I don't know what. So let's focus on organellar contacts. So how to check this? Well, one of the method is to use there are two methods. One is using microscopy, and the other is using biochemical tests. And microscopy is a kind of subjective, as it depends on the resolution of the microscope. If the resolution of the microscope is high, if the resolution of the microscope is low, then two points which are far away, they can look to be the same point. So even if two organelles do not contact each other, even then it might look that they're contacting. So a better resolution is needed in this case, and different results will be obtained from different microscopes with different resolutions. And the second is the biochemical test, which is much more objective. An example of this is PLA assay, or PLA, that is proximity ligation assay. Now for example, we want to test the contact between two organelles, for example mitochondria and ER, or endoplasmic reticulum. So the PLA kit of this would contain first of all primary and primary antibodies against against a particular protein which is very common in mitochondria and ER, and their secondary antibodies would be out there. To the FC region of the secondary antibody, there will be a single-stranded DNA which is 20 to 30 base pairs long. Then the other components in this kit of PLA would be single-stranded circular DNA with certain portions which are complementary to that of the secondary DNA attached to the secondary antibodies, because if the organelles are close enough, then there will be certain region where let's imagine two spheres are coming in contact. Then there will be certain region which is from the surface of one sphere to the other, which is around 40 Armstrong or 40 Armstrong, because even after contact there is certain points where they are separating, right? Since this is spherical, to show it, if this is one. So if this is happening, so this is the closest part, and there's certain part which would be 40 nanometers. So there it would, this antibody thing would be binding, and then to the single stranded DNA which is attached to the single stranded DNA which is attached to the secondary antibody would complementarily base pair with the single stranded circular DNA, because it has complementary regions, right? So, and there will be a polymerase in that kit which would be replicating the other regions which are not which are not this part are key, since these are not yet having, these are still single stranded. And there in the kit there is a probe which binds to the double stranded DNA. So I mean it would bind to this double stranded DNA, and so if this whole structure is formed, if the this it will be formed when if the two organelles would be coming closer or close or in contact, or if they are closer than 40 Armstrong, as this whole region would be 40 Armstrong. So then this this probe would be able to bind to this double stranded DNA, as only then the double stranded DNA would be formed, and we would see some fluorescence. And this is it. And there is another hypothesis that this 40 Armstrong nanometer is formed as the organelles which touch each other, there is some lipid membrane fluctuation which results in this 40 nanometer distance between each other, and this is it. So let's recap a little bit of this. That what does the PLA kit contain? Well, it contains the primary and secondary antibodies to which and against a common protein. The primary antibodies against a common protein of the two organelles, and there are secondary antibodies for the two and primary antibodies. And to the Fc region of the secondary antibody there is a 20 to 30 base pair single stranded DNA which would complementarily bind to a plasmid, and there is a DNA polymerase which would be replicating the rest of the region, or polymerizing the rest of the region and making it a double stranded circular structure. And so the probe that has affinity for double stranded DNA would be binding to it and resulting in fluorescence or something like that I don't know. And that's it.